Hi, this is Miss Litton, and this is the beginning of our discussion on Chapter 42, Animal Development. And we're going to go through and hit some of the key events that happen during development. We're going to look at some conserved properties between other organisms and our own development. And basically, we want to know how we get from here to here. And a human develops from a single cell. That always is amazing to me. You are all one cell. One cell, and look how beautiful you are now. And you are trillions of cells all working together, right? So we talked about this already, about fertilization and implantation. At this point right here, getting ready for that egg to pop out of the ovary. Do you remember what hormone causes the egg to pop out of the ovary?
No.
LH. Think leave. Okay? So luteinizing hormone causes it to pop out, and that egg is only viable for maybe 12 to 16 hours. And here's the deal. You could have sex on Monday and fertilize an egg on a Friday.
But it's still there.
Yeah, what happens is the sperm that is in the oviduct can just be sitting there waiting, for an egg to come out, and then all of a sudden the egg comes out, and then game on, okay? So boom, here's a little bit of ovulation. Out comes the egg, okay? And so the sperm need to be present, and it needs to be in the oviduct in order to fertilize that egg, because by the time the egg moves down and closer to the uterus, that egg is no longer functional. And it's going to spend about 4 or 5 days traveling from the ovary all the way to the uterus. So sperm have to be right around that time when you're ovulating. So on the top of each sperm, they're wearing a cute little cap called an acrosome. And that acrosome, that looks like a little beanie on the sperm's head, it contains enzymes to get you through the zona pellucida, which I think sounds like, did I tell you this before? To me, it sounds like a summer concert series with a bunch of girl bands, Zona Pellucida 2017. And the enzymes in the acrosome will digest through that. Now, there obviously is only one winner, right? Winner, chicken dinner here. Only one sperm can get in there, and they're all vying to be first. And it's kind of incredible. We'll talk about it. There's a fast and a slow block, but when one of them penetrates the membrane, when just one of those sperm, it causes a depolarization, just like on a what? Where did we see depolarization before? On a action potential on a neuron. Good, just like that. Okay, so it has to digest through the zona pellucida, it has to fuse with the plasma membrane, and then penetrate that egg. So here we can see one doing just that, and let's watch the sperm cam. Sperm cam. That's probably good. I know I won't get that far. Thank you so much. Super appreciate it. That, my friends, is a white blood cell. Swim, little sperm. Watch the music. Get ready for the GoPro.
It's like a torpedo.
the sperm nucleus. He's got to fuse with the egg nucleus. It's powerful. There it goes. Tinkerbell like. Yeah. All right. So that all has to happen, just like that. All right. So, yes.
Why did that one make it, not the other one?
Well, they're all working at it, so one of them just gets there faster. I think they just did that to make it dramatic. Yeah. So immediately the egg forms a barrier to prevent other sperm from entering the now fertilized egg, because you don't want to be a mutant Ninja Turtle. So there is what's called the fast block and the slow block. So here you can see the acrosome right here, this little cap, and it's releasing those enzymes. This band right here is the zona pellucida. This right here is the corona radiata. There it is. Corona radiata right here. This comes from, remember how the egg matured in the, do you remember what it was called? F, starts with an F. Egg matured in the follicle. Those are residual follicular cells kind of around it. That's the corona radiata. And then here you can see the zona pellucida. And now the sperm membrane and the egg membrane have joined, and you're going to fuse. And a haploid plus a haploid makes a diploid zygote. Good. Okay. And so here you can see that process. You can see the corona radiata. You can see, and the tail doesn't come in. It's just the sperm nucleus. And so then your mixing of the chromosome and fertilization is complete. And then development, what this chapter is about, is all the changes that occur in your life cycle. Not just up to when you're born, but as we continue to develop and break down as we age as well. So on your notes, go to details of human fertilization. You have one already, sperm bores through remaining follicular cells called the corona radiata. The acrosome is a cap on the sperm that releases enzymes that digest the zona pellucida, the jelly coat, peanut butter and jelly. Why doesn't slate do 3 and blue do 4? Go ahead. Okay, guys, don't watch the link now, but you can watch it later. There's like a little 10-second video. It's pretty interesting. The fast block is as soon as the sperm membrane touches the egg membrane, it causes a depolarization, similar to a neuron. And then blue, why don't you explain the slow block? All right. So let me teach you something now. Okay, make an egg. And I think I teach this part to honors. I think there's a video of me doing this already online. I think. I'm not 100% sure. On the other hand, needs to be a sperm. Here comes Mr. Happy Sperm. Here we go, right here. With me. Everybody's here. Here comes Mr. Happy Sperm. Okay? And he's going to use the, what's on there? The cap. What's the cap called? Acrosome. And so it can penetrate the egg, and then you form a diploid zygote. Here comes Mr. Happy Sperm, and he uses the acrosome, the cap, and in order to penetrate the egg, and you can form a diploid zygote. Now, this zygote right now is going to do mitosis, right? Right? Because we're diploid, not meiosis. Mitosis, and it's going to do it so quickly. Repeated mitosis. It's not going to go through G1. Remember what G1 is?
Yes.
Growth and the organelles double. Remember all of that? Yeah. Now, the DNA is going to replicate. You're still doing S, G2, but your G1 is like non-existent. So when you do this, this would be one cell. In the same amount of space, you'll have 2 cells. In the same amount of space, you'll have 4 cells, 8 cells, 16 cells. It's not doing growth, so they don't get any bigger. And this stage, this where you have this repeated mitosis, is called cleavage. Okay, and you remember
Cleavage furrow.
cleavage furrow. So you're going to go like this. Heavy sperm, diploid, zygote. Go, mitosis, mitosis, mitosis. Mitosis, mitosis, mitosis. And then cleavage, cleavage, cleavage. So you do a whole bunch of cleavage, and you go like this. Watch me. You form a solid, little circles, a solid ball of cells called a morula. I'm having you learn it in obnoxious ways so that you will remember, right? Here we go. A solid ball of cells called a morula. Good. Now, this is fairly clever. You're going to pump in salt to the center. So pump 2 times. What are we pumping in there? Salt. We're now making the interior what? Hypotonic? What? Hypertonic. Who's going to follow that? Water. And this will blow it up like a water balloon. You're going to form a blastula. So now we're a hollow ball of cells. Okay, so go right here. Sperm fertilizes. We form a diploid, zygote. Cleavage, cleavage, cleavage. We are a solid ball of cells called a morula. Now we're going to pump in some salt. Who follows it? Water. And you're going to form a blastula, which is a hollow ball of cells. This cavity inside is called a blastocele. Say it. Blastocele. Now, we are a hollow ball of cells. Do you remember those red rubber playground balls that you had when you were a kid? Yeah. Now, let out the gas of it. Like, if you let the gas out of it, okay, like you wore it as a hat or something, okay, you let out the gas, okay, this is forming a layered ball of cells. Go ahead. Layered ball of cells. It's called a gastrula. Gastrula. And I think letting out the gas, okay? Now, this layered ball of cells, initially, you're just going to have ectoderm and endoderm, but then you'll form the middle layer, which is the mesoderm. Good. So take a look up here. Now, I'm showing you a lancelet of fish, which has very little yolk. Okay. So here we can see cleavage, repeat mitosis. So it's about the same size, but now we do cleavage, cleavage, cleavage. We form a what? Solid ball of cells called a morula. Okay, the morula is a solid ball of cells. We're going to pump in some salt. Who follows? Water, and we form a blastula. Okay, and a blastula is a hollow ball of cells. The cavity inside is called a blastocele. Now, within this blastocele in humans, this is not human, okay? But in humans, we form what's called this inner cell mass, and the inner cell mass is actually going to be the baby, okay? For us. So we don't see that here. This folding that you see right here, you're forming a gastrula, which is a layered ball of cells. Okay. Now, from your layered ball of cells, we're going to develop ectoderm and mesoderm and endoderm. And from the gastrula stage, the last stage that we cover on this is called the neurula. And the neurula is when the nervous system forms. But I haven't got to that, but I wanted to hit that stage name with you. Okay, on your notes, go to your notes. Embryonic development. Do you want a picture of this? Is that what you're hoping for? Okay. Embryonic development. The morula, what are you going to put right there? Solid ball of cells. As a result of repeated cell division without growth. Blastula is a hollow ball of cells, and the blastocele is a cavity inside the blastula, and the inner cell mass, the ICM, develops into a baby. The tissue stage of development is a gastrula, and this is a what ball of cells? Layered. Yeah, layered ball of cells. And when you form that layered ball of cells, you are going to get 3 layers ultimately. And that's going to be the ectoderm, the mesoderm, and the endoderm, and that's where we'll pick up next time. Good job, guys. Hope that's helpful.